Hello children, today I am going to discuss with you the last lesson of your grade 10 book. It is inheritance. Under this lesson we are going to discuss what are the inherited characteristics and how they are inherited from generation to generation. First of all, let's see what are the inherited characteristics. Inherited characteristics are the characteristics or features of the organisms that is passing from one generation to another generation when they are doing reproduction. There are two types of inherited characteristics. One is common inherited characteristics, other one is rare inherited characteristics. Let's see what are the common inherited characteristics that can be found in human beings. Skin color, curly and straight hair, fuse or free earlobes, ability to fold the tongue, position of the thumb when their fingers are crossed, dimple and normal cheeks, straight and curved thumb, widow peak on the forehead. Those are some examples for the common inherited characteristics. Some of them can be seen in this figure as well. Then next one is rare inherited characteristic. As you can see in the figure number 1, it is the syndactyly. Syndactyly means joining the two near fingers in its skin to some extent. The second figure you can see it is albinism. There is a extremely fair skin, white hair and white eyelids when they have in albinism because they doesn't produce melanin pigment their skin. The other one is polydactyly. Polydactyly means presence of 6 fingers in the hand or foot. And there is a other one too which cannot visible in this figure. It is brown or blue eyes. The experiment about the inheritance was done by the scientist Mendel. Mendel was selected pea plant for his experiment. Let's see why he selected pea plant for their experiment. Because it can be easily grown, can obtain yield within a short period of time, can obtain pure breeding plants. There are easily observable contrasting characteristics. I'll explain what are the contrasting characteristics in later. And naturally self-pollinating when necessary. Cross-pollination is also possible. And also ability to get offsprings by breeding and can continue the generation. These are the reasons why he selected pea plant for his experiment. At least 2 or 3 you should have keep in mind if the question is asked in the question paper. You have to write at least 2 or 3 of reasons. Let's see what are the contrasting characteristics of pea plant. Contrasting characteristics means the opposite features that can be seen in the pea plant. There are several contrasting characteristics when considering the seed shape. The pea plant has round and wrinkled seeds. When considering the seed color, yellow-green color, and also flowers has purple and white color. Pod shapes, inflated and constricted pods, and the pod colors yellow and green, and the flower position axial and terminal flowers. And the last contrasting characteristic is the stem height. It's tall and dwarf. For these contrasting characteristics, the Mendel was done his experiment. Before moving to draw the genetic maps, let's move into the basic concepts of genetic. You have to know this basic concept, and some of them are most important, so keep in mind these definitions in your mind. First is monohybrid cross. Monohybrid cross means consider a single pair of contrasting characteristics at a time in genetic experiment. For example, when we are doing a genetic experiment, we have to select one characteristic at a time. For example, for the pea plant, we can take tall and dwarf in one time. In second experiment, we can take color of the flower. Likewise, you have to take the single characteristic for one time. The second one is the dominant character. Dominant character is most abundant feature. When we are drawing or when we are writing a genetic maps, we have to represent the more dominant character by capital letter. The recessive character is the character which is rarely found. It is represented by a simple letter. We have to select the relevant simple letter of the dominant character. If you are going to select capital R for the dominant character, for its recessive character, you have to select the simple R. The next one is homozygous. Homozygous means two genes are similar for a given feature of an organism. You know, we have genes which is corresponding to our features. For every gene, there are two for every character, there are two genes in our DNA molecule. If they are similar, it is called homozygous. For example, capital R, simple R, simple R. They are homozygous. The heterozygous means the two genes are not similar for a given feature. One is capital letter, the other one is simple letter. When there is a heterozygous feature, the dominant character will appear externally. The phenotype. Phenotype is the feature that externally appear. For example, color of the pod, height, and the shape of the pod. Those are the externally appeared feature. Genotype is the gene composition to determine the phenotype. For example, the red color can be red color has gene composition capital R or capital R simple R. Likewise, the gene composition is the genotype. Then next one is the gene. Gene is the specific base sequence in a DNA molecule that represent a particular character. You know that DNA are the molecules which storage our genetic informations. The genetic informations are storage in as a base sequence. They are called genes. Gene linkage. The gene that represent in the same chromosome which are not segregated independently. You know that when we are doing reproduction, we produce gametes. For example, males produce sperms. When they are producing sperms, their number of chromosomes will be getting half to produce the sperms. That means the 46 divided into 2. So sperms get 23 chromosomes. These chromosomes are divided equally to each sperm cells. But some genes are not segregated equally. That is called gene linkage. The next one is heredity. Transmission of inherited character to next generation. For example, our skin color can be transmit into our offsprings. If you are having a fair skin, your daughter or your son will have fair skin. That is heredity. The inheritance means the process of transmitting inherited characteristics. Let's see how to inherit characteristics from generation to generation by doing monohybrid cross between contrasting characteristics of pea plant. First, I have chosen the color of the petal. As you can see in the figure, first one has purple color petals and the second flower has white color petal. When we are doing the cross pollination between these two flowers, the first generation will have purple color petals. That means dominant character. This F1 generation or first generation will appear dominant character. Dominant character will appear in first generation. In here, the dominant character is purple color. Right? Let's draw the genetic graph for this one. I'll take purple color as dominant character. I'll represent this dominant character by capital R. So as you know, for every character there are two genes. So purple color has capital R. For the simple white color has the recessive one has simple R. By crossing between them, capital R and simple R, you will get capital R, simple R. This is the heterozygous feature, but externally feature purple color, that dominant color. This is the F1 generation. If you want to take the F2 generation, that means second generation, you have to cross capital R, simple R flower and capital R, simple R flower together. Capital R, simple R pollens and capital R, simple R ovules. When we are crossing capital R, you will take capital R once, and the capital R and simple R, you will get capital R, simple R, and the capital R, simple R, again capital R, simple R is white color. Finally, you will get 3 purple color flowers and 1 white color flowers. That is the 3:1 ratio. Let's say take another example. It's the tall and dwarf. If I selected parental generation tall and dwarf, the tall is the dominant character. It is capital T. Dwarf is the recessive character, simple T. Gametes have capital T and simple T, and after fusing these gametes, first generation get capital T, simple T. Those are all of them are tall. That is dominant feature. Always the F1 generation will get dominant feature. After self-pollination of the dominant flowers, F1 generation, in between F1 generation, you will take gametes capital T, simple T, capital T, simple T. By crossing these two, you will get capital T, capital T, capital T, simple T, capital T, simple T, and simple T, simple T. Capital T, capital T means tall. Then capital T, simple T also externally appear as tall, and the simple T is dwarf. Right? If you want to write the phenotype and genotype in here, you can see there are 3 tall plants and 1 dwarf plant. Phenotype is externally appear feature. So you can externally appear tall plants and dwarf plant as 3:1 ratio. It is phenotype. For this one, genotype is the gene composition. You can get capital T flowers, capital T simple T flowers and simple T flowers. 1:2:1 ratio. It is the genotype. You have to write as a ratio when you are writing genotypes and phenotypes. Another example is the pod color. The pure yellow pod color has capital Y. Pure green flowers is simple Y. In here you can see from the letters, pure yellow is the dominant character. The gametes will receive capital Y and simple Y. After fusing them, F1 get capital Y and simple Y. When doing the pollination between F1 generation, you will get finally capital Y, capital Y, simple Y, capital Y, simple Y and simple Y plants. The ratio between pure yellow to pure green is 3:1. Pure yellow to pure green is 3:1. It is the phenotype. The genotype ratio is capital Y to capital Y to simple Y 1:2:1. It is the genotype. Then let's see heredity of human. You know that the human there are 46 chromosomes in their somatic cells. From these 46 chromosomes, that means 23 pairs, as you can see in this figure, there are 22 chromosomes are somatic chromosomes, and the one pair of chromosome is considered as the sex chromosome. 22 pairs are autosomal chromosomes and one pair is considered as sex chromosomes. In females, their sex chromosomes are capital X. For the male, they have capital X and simple Y chromosome as the sex chromosomes. It is the sex chromosome. Sex chromosomes. Other 22 pairs are considered as autosomal chromosomes. That are the chromosomes which determine the features of your body. Then let's see how to determine your sex from genetic map. Let's take male and female. The male has their genetical composition XY and the female consists XX genetical composition, genotype. When they are producing sperms, males produce X and Y sperms and female produce X and X eggs. When they are fusing together, the offsprings get capital X, capital X, capital X, capital X, capital X and simple Y, capital X, simple Y. That means XX means girl, boy. The genotype ratio is capital X, simple Y, 1:1. 2:2 means when we are simplifying 1:1. The phenotype is girl to boy, also 1:1. That means there is a 50% probability to get male boy and 55% probability to having a girl. Then let's see how to inherit human genetical disorders. Under this, we are going to discuss two types of genetically disorders. One is the disorders due to sex-linked recessive genes. That means because of having simple letter. First one is hemophilia. The hemophilia is the disease which occurs due to a X-linked recessive gene only present in male in population. It is so important. It is only present in male. The females are carriers. Let's see how to inherit hemophilia. There are two examples I have got in here. First one is if the mother is carrier, X is a carrier and the father is healthy. This hemophilia is get because of simple letter, no? So I'll get the healthy one as capital H, the disease one as simple H. So carrier's mothers has one capital H and one simple H. One capital H and one simple H, because she is carrier. If she is healthy, both of chromosomes are capital H. If she is unhealthy, she has simple H. Carrier means one is dominant, other one is recessive. The healthy male consists capital H only. Then after crossing these gametes, first female will get capital H, that is, so first one is female, the second one is male, XY, third one also female, and next one male, right? The first female will get this one, capital H, simple H, and second female will get capital H. The next male will get capital H and Y. Next one will get simple H and Y. Then this is the gene composition, genotype. The phenotype will get carrier daughter, affected son, unaffected daughter, and unaffected son. If the mother is healthy and the father is unaffected, the gene composition will be capital HX and capital HX, and the father will simple YX and simple X, simple Y and X, simple H and Y. As a result of this, they will get, the offsprings will get capital H, simple Y, capital HY, capital H, simple YX, and capital HY. Carrier daughter, unaffected son, carrier daughter, unaffected son. Then let's see the another sex-linked recessive gene disorder. It is red-green color blindness. When suffering a red-green color blindness, they cannot distinguish red color from the green color. This is also common among males but rarely occurs in female. Hemophilia is only present in the males. The carrier ones are females. But red-green blindness, there is a common feature among the males but rarely occurs in females. As you can see in the figure, the red-green color blindness also can inherit from generation to generation. If the female is a carrier, she has capital B and simple B allele genes, if the male is healthy, he has capital B and simple Y genes. As you can see, the offsprings will get unaffected female, carrier females, unaffected males and color blind male. The next disorder type is genetic disorders that occurs due to gene mutations. If the mutation occurs in the genes of producing melanin, they will suffer from albinism. It is extreme white color or extreme fair color. They have extreme fair color skin, hairs and other external features because of doesn't producing melanin pigment. The other disorder is thalassemia. When having thalassemia, their red blood cells has disorder when having thalassemia. Right? The final topic of this lesson is genetic engineering. The genetic engineering is applied in different areas, as you can see figure, in industries, medicine, environmental and agriculture field. I'll explain first what are the applications that apply genetic engineering for the agriculture field. When having agriculture field, the genetic engineering is applied to produce weedicides resistant crop and pest resistant crops, to produce rice enriched with vitamin A, that means golden rice, and also a tomato resistant to cold weather, and to produce high productive cattle and milk with high nutritious value. For the industrial field, genetic engineering is applied to production of enzymes such as amylase, to produce some amino acid, for example monosodium glutamate, and also to produce vitamins by cyanobacteria, vitamin B12 and E, and as a remedy to the pollution by fossil fuel combustion and other waste material. For the medical field also applied genetic engineering for the production of insulin from E. coli bacteria, and for the production of proteins including growth hormones, and production of antibiotics when repairing organs, for the gene therapy, and to use DNA technology to confirm the identity of the person in forensic medicine. Those are the areas which apply the genetic engineering. This is the end of your grade 10 last lesson. I'll continue this session for the grade 11 too. Let's meet from grade 11 lesson soon. Until then, goodbye. Don't forget to subscribe my YouTube channel.